We're so excited to hear from Dr. D'Angelo today as he helps us understand nutrition and the gut microbiome and the role they play in multiple myeloma. We chose today's topic because understanding the gut microbiome and how it affects our bodies has become something that a lot of us have been trying to understand. I also think it helps empower people in taking charge of their care. They may not have a lot of control around what treatments it will be next or how they'll react to that treatment, but we decide daily what foods we eat, and knowing that it could be affecting your multiple myeloma can give you some of your power back. And in talking to Dr. D'Angelo before this presentation, his feelings mirror that as well. So with that, I'll go ahead and introduce today's speaker. Dr. Christopher D'Angelo is an assistant professor in the Division of Hematology and Oncology at the University of Nebraska Medical Center. After obtaining his bachelor's degree from Northwestern University, Dr. D'Angelo attended medical school at the University of Illinois College of Medicine, graduating with AOA honors. He completed a residency in internal medicine at the University of Chicago Hospitals and Clinics and a fellowship in hematology and oncology at the University of Wisconsin. Dr. D'Angelo treats patients with lymphoma, multiple myeloma, or non-malignant blood conditions. He specializes in stem cell transplantation for these diseases, as well as cellular therapy options, including CAR-T. He has special interest in the gut microbiome as an important immunological environment and how that may affect the hematologic malignancies. And with that, Dr. D'Angelo, I will go ahead and turn the time over to you.
Wonderful. Thank you so much for that introduction, and thank you to everyone who's attending this webinar today. Just bear with me as I get my screen going here. Okay, I think that's the way it should look. But anyway, yes, thank you all for taking the time to come today.
Sorry, Dr. D'Angelo, I apologize. It actually has your next slide showing as up. It's a little different than earlier.
Oh no, okay. Okay, hold on, we'll try this again.
Sounds good.
So is that better?
That's perfect. Thank you so much.
Gotcha. Thank you. So, okay. So anyway, thank you all for attending, taking the time to talk today and listen to this discussion, and appreciate your interest in trying to understand the role of the microbiome and the role of nutrition as we're thinking about cancer care with specific respect to multiple myeloma. I want you to know I'm going to start this talk with kind of a general discussion of, I think, what the power and the hope of nutrition can do in exploring outcomes and ways to improve their care in myeloma, discussing a little bit of research on patient interest when it comes to nutrition, and then kind of pivoting and moving a little bit further into the microbiome and nutrition, particularly with respect to multiple myeloma and some of the therapies that we use to treat. So to get started, I do need to disclose that artificial intelligence was used to create this presentation. But I promise it's only for the little pictures that I've created here, because I couldn't help myself. If you guys haven't had the chance to try it out yet, you could try using the Google AI device, like the little extra thing, or Bing search AI, and ask them to create your prompt for you. And in preparing for this talk, I said, you know, can you make me I asked the Bing agent, can you help me make a picture of using nutrition as kind of a weapon or a fighter in as it comes to cancer care and the microbiome as well. So after a few drafts, and you definitely have to work with it a little bit, it came up with this first figure on the left here, where we've got our cancer team here, and then we have this little broccoli fighter here, which I just kind of loved. I thought he was awesome. And he's the sort of prebiotic that we use in our treatments when we're thinking about nutritional interventions. So I tried to build on him, directly kind of engaging with cancer in this middle figure here. We've got our broccoli doctor here going to battle, you know, the cancer. Not that I mean to use that type of language, so forgive me for that. But finally, I wanted to get the microbiome into the picture here too, and so you have the cancer hero. And then the microbiome kind of best buddy in the background. That's kind of the gentle giant. That's maybe the hope of how microbiome research can work here. So thanks, and forgive me for having to discuss my art here and stuff. I appreciate you guys bearing with me on this. So moving forward, these are a little bit of my conflicts of interest. I promise none of these are really relevant to the discussion today. And what I really want to do is discuss the gap between patient interest in nutrition and oncology recommendations. Many of you are probably more than aware of this gap or have lived it personally. So if you have, I want to empower you to bring this up and to support you in knowing that you're not alone in this, and that I think these next several years will be a bit of a change, as you can see oncologists pivoting more into having these discussions, particularly if we're following our patients' lead on this. I'd like to follow up with some simple nutritional objectives and recommendations that we can share during cancer therapy, and then understand the role of the microbiome and its impact in oncologic disease and therapy. And most of this will be myeloma focused. So my focus is in lymphoma and multiple myeloma. And then, in conceptualizing this interaction between nutrition and cancer, I kind of came up with 4 different ways that this could engage. There's a hope that nutrition can help prevent cancer. There's a hope of nutrition that it can be used to help treat cancer, to help support some of our patients through cancer as they're dealing with side effects, or how we can optimize those other things with respect to their comorbidities. And that's kind of what I was trying to outline here, and a couple of different ways that we could do that. Supporting patients through malnutrition, for example, is one key and more direct connection that nutrition can play. But I also think it can help treat, particularly potentially via micronutrients or the microbiome, which we'll spend most of this time discussing. And so first, I want to highlight some work on the patient interest. This is a paper that was published by a good friend of mine, Dr. Shah, who's a myeloma and nutrition specialist at Memorial Sloan Kettering. And she conducted this work with her team, which was actually supported by the HealthTree Foundation and some of the data that they have generated. And I think they came up with a really neat study here to just draw some attention to this need. This is a survey of patients with plasma cell disorders, so multiple myeloma, and then the sort of precursor diseases that can come along with this. And we obtained about 400 subjects. So when it comes to nutrition and diet, there's a couple of questions that came up here. And one was just simply, since you've had a diagnosis of myeloma or a related disorder, have you had questions about your diet and nutrition? And then how best did your oncologist address this? And you can see there's some data on the right here. And what I want to highlight here is that 82% of patients have nutrition questions. This is a very common question that comes up here. And despite that interest, only half of them are being met by and addressed by cancer doctors. And so when you dig into it a little bit more, what advice did you receive from your hematologist and oncologist? You can see a couple of different options there. Eat more plant-based foods or fiber, eat less junk, eat a balanced diet, be referred to a nutrition, or no advice at all. And unfortunately, boy, that no advice at all, not even a referral to nutrition, was observed in about 44% of patients reporting in this. And so certainly, kind of addressing an area where we could improve. But when they did get advice, 29% got this balanced diet. And honestly, I've even been guilty of saying this sometimes in clinic. What the heck is a balanced diet to begin with? Is it balanced with what? Balanced with meat and veggies? Is it balanced with all of these things all together? And does that diet work for me and my myeloma? Does that work for my partner and his diabetes? Is there a right tool for the right job here? And I certainly think that there probably is. So this balanced diet, I think we could do a little bit better than just saying eat a balanced diet. And that's kind of what we're highlighting here. And only 25% in this group was referred to a nutritionist. So certainly an opportunity, particularly if you're engaging with your cancer team and stuff like that, to ask if that is an option that's available. And so finally, would you like your oncologist to make recommendations? And are you interested in changing your diet? 66% of survey participants said they would like their oncologist to provide some recommendations. And 82% would change their diet potentially based on these recommendations. And so the take-home message from this data, at least my take-home message in reading it, is our patients are interested in dietary advice during their cancer treatment. They're looking to their oncology providers for leadership and are even likely to follow the advice that comes from that. I think I certainly recognize, the impact that providing good guidance on can have for our patients, particularly in a realm that is garnering far more interest in recent years than before. So I think certainly room for improvement and ways that we can think about how we do this moving forward. I couldn't help in researching this talk find a quote that was taken from a cancer paper in the 1980s, because in many ways, much has changed and yet much remains the same. And what I wanted to highlight here, first, the title was just Faddism and Quackery in Cancer Nutrition. And what I want to point out here is that, I mean, these numbers have changed, but they're writing 50,000 cancer patients annually, pockets picked by promoters of nutrition quackery, meaning those that are advising kind of snake oil, for lack of a better word, or treatments that probably cost a lot of money but have really no proven benefit. And he points out here, which I think remains true, why does this go on? Well, unfortunately, we need to recognize that our patients are susceptible to this, especially when they feel hopeless and not in control of their therapy. Hopeless, helpless, and not in control of their therapy. And why does that happen? I think we recognize that cancer has a way of taking control from a lot of patients. You know, their lives suddenly revolve a little bit more around showing up to see their cancer doctors, getting their treatment. If they don't show up and get their labs, or if they don't show up and get their treatment, they could be jeopardizing their cancer outcomes. And it can certainly have a way. I'm not necessarily saying that that's the case, but I can understand if those are the thoughts that circulate in our patients' heads. And they can have a risk of snowballing, and you can imagine the vulnerability that comes along with that. In what can rapidly become a feeling of not being able to control nutrition, and the things that we put in our bellies every day is actually one thing that our patients still can have some control over. And so you can see naturally where these questions come from. And I think the next point that I want to point out is there's a significant patient interest in this. And if we don't have matching oncology input, there's a deficit that can be created here, a power void that can be filled by opportunists that are exposing our patients to harm, particularly if they're promising something that sounds too good to be true and offering a chance at control that might not have been weighed in on by the oncology team taking care of our patients. So I just want to recognize that this exists, and that if we can define it and name it, perhaps we can start studying it and think about ways that we can improve it. So how can we do that? Well, one easy thing that we can do is American Cancer Society has guidelines for diet and physical activity for cancer prevention. I will say some of these can be cancer specific. Some of them are kind of larger, just general recommendations, and I don't think any of them are in any way harmful. So I know that this is something that's written a little bit more for maybe a clinician-based office, but your library might have access to this, and it has some nice little bullets on how you can take a look at them that I think are reasonably patient-friendly. One example with respect to prevention is whole grains. Here, this paper kind of noted that 30 grams per day, which is just probably a couple of servings, can translate into a 5% risk reduction in colorectal cancer. This is not to take this too far into colorectal cancer research, but this is certainly something that is gaining interest as younger patients are being diagnosed with colorectal cancer. And how can whole grains work? Well, they're rich in these so-called phytochemicals, which can have metabolic activities that extend beyond just nourishing your own microbiome. They're rich in fiber, and all of this translates into reducing gut transit time. And so if your gut is moving things through a little bit faster, and there's certainly a sweet spot for that, you don't want things to be too fast, but you don't want them to be too slow. And that can lead to potentially less exposure to carcinogens. If we've taken things in and are processing them slowly, your gut has more opportunity to interact with these. But if you've kind of bound them up with fiber and some of these other nourishing compounds and nutrients, that might increase your gut transit time, and could also be beneficial for microbes. The evidence base for here I would grade as probable. I think it probably works, but to be honest with you, this data mostly comes from what we call epidemiologic surveys or studies, where we ask patients to recall what they ate and kind of record that, and it's not as perfect as taking people, recruiting them to a clinical trial, and then randomizing them to a diet intervention and seeing what comes of that. That's the level of evidence we need. And that's the level of evidence I think we're starting to build as we think about cancer care, nutrition, and then another environment, the gut microbiome. And so pivoting a little bit more into prevention studies for myeloma, it helps to know that there is some data that's out there, even if it is of not perfect science when it comes to epidemiology. There is data here, and it gives you some food for thought, no pun intended, or maybe pun intended. But this was titled, Dietary Intake is Associated with the Risk of Myeloma and its Precursor Disease. This is a study that was conducted in Iceland, or at least with authors that are Icelandic. And I mentioned that because they're doing a very cool trial right now where they are screening all of the people that live in Iceland for plasma cell disorders. And by doing that, they have this population that they will get a very rich data set on how many people have this MGUS precursor myeloma disease, how many of them go on to develop myeloma, and maybe even better understand why they go on to develop myeloma. In particular, asking potentially some questions like environmental factors, like the food we eat. And going into this a little bit more, this was a survey of 5,000 people, including 300 cases of MGUS, that precursor multiple myeloma disease, and then multiple myeloma itself. And what they saw, I think, is at least what I call hypothesis generating, which should turn your head a little bit and wonder, maybe there is something here that's worth going into. And that's how I interpreted this. They noted that if you had a greater intake of fruit as an adolescent, you had a reduced incidence of MGUS later in life. That's important because MGUS is the sort of precursor disease to multiple myeloma. Almost everybody has an MGUS period before they go on to develop myeloma. And I compare it to it's kind of like the colon polyp of colorectal cancer, except you can't just remove the polyp like you can when it comes to colorectal cancer. When you look at adults with MGUS, if they had an intake of fruit that was above kind of in the higher quartiles of fruit intake, they had a reduced risk of developing myeloma. And these odd ratios of 0.34 and 0.62 are not only statistically significant, meaning likely meaningful, but that's a pretty good protective benefit here, and certainly something that should, I think, raise more attention in this research space. Finally, I have mentioned a lot about the limitations of survey research. It's really good, but has some limitations to it. The sort of gold standard when we talk about research in medicine is clinical trials. And so what I'd like to highlight here is a clinical trial on nutrition support in patients with cancer that were admitted to the hospital. So this is all cancer patients, not necessarily blood cancer or myeloma specific, but I still think holds some truth in thinking about myeloma specific endpoints. And so this was called the EFFORT trial, which was a randomized controlled trial, or an RCT, that was conducted in Switzerland. This randomized oncology patients that were screened for nutrition risk and randomized them to a dietitian-directed nutrition intervention and support, or just kind of standard support where they just ordered their food tray as always. This was published in The Lancet, which is a very, what I call, high-impact journal in 2019. And basically what they took a look at was individual nutrition targets. They looked at caloric requirements, protein requirements, micronutrient requirements, and then specific targets that were meant for specific adaptations, like low potassium in patients with renal failure, for example. And they had a couple of kind of levels to it. Their first level was to just feed the patients meals adapted to preferences, or could they be enriched, and could you do snacks in between mealtime, and multivitamins too. They would then reassess in 1 or 2 days if they were not hitting those endpoints that they had highlighted before, the amount of calories or the amount of protein. Then they added kind of a level 2, and the level 2 was enteral nutrition, using a sort of food tube-based kind of approach to really optimize nutrition on the inpatient side. And if that failed, they escalated to level 3, which is nutrition through the IV. So really kind of emphasizing nutrition at multiple levels and continuing those levels. Even if you went on, you didn't just stop your oral food intake, you continued it and then added on the extra layers of support. And so when they took a look at outcomes, what I'm showing here is a curve where, as the curve gets lower, these people are unfortunately not surviving or dying from this. So the lower it goes, the worse things are. And you can see here the maroon curve at the top here that shows individualized nutrition actually had less deaths compared to the usual care at 30 days. So I'd like to say this again because it's important. Just using a simple dietary-based approach was powerful enough to improve people's lives and help them live longer at 30 days. It reduced mortality at 30 days. And I can tell you guys, if this weren't a nutrition intervention and this was a cancer drug, you can only imagine the amount of resources and input that cancer doctors would be using and how much we'd be trying to use this in the clinic. And what's even better is it's not a drug. It doesn't really have, those kinds of drug side effects. It's nutrition, and we can use this. And I think that the conclusions that I want to draw from this is that nutritional interventions can be potent. This isn't just a sort of side quest, for lack of a better word. This could be the main event here, at least, in conjunction with other therapies. And it can be potent. Even with that dietitian-based intervention, 73% of patients met goals. If that only worked with 73%, you can only imagine, is the sky the limit here? What can we do to increase that intake even more compared to the 22% that just got their usual care? So you can see not only did it impact far more than not having that intervention-based guideline, but I think there's even room to get even better on this. This is also sort of a shout out to our dietitians that support our cancer patients. They do great work here and need to be involved even more often. If a little is good, I think more is better in this scenario here. And so continuing this kind of paradigm on the hope of nutrition, what I want to pivot next into is the microbiome, where I want to spend the rest of the time talking about this a little bit more. And the way I was kind of conceptualizing this is nutrition is the gear that can turn the microbiome, which is the gear that might be able to turn cancer care to improve outcomes here. At least this is kind of how I think. I also think nutrition doesn't just indirectly impact cancer care through the microbiome. It also probably directly impacts this wheel as well, but just kind of these ways in which this can kind of influence and work together here. And so, if you have heard about the microbiome, or if you haven't been, you can certainly appreciate the fact that it might be having a moment here. This is a paper from the New England Journal on microbiome predicting mortality in patients receiving stem cell transplants for blood cancers. This is microbiome associated with cognitive function in the midlife. This is the microbiome affecting the intestinal barrier and associated with anxiety and depression. This is the microbiome in cardiometabolic disease, the microbiome in liver cancer, diet and probiotics in melanoma immunotherapy responses, bacteria in the microbiome in multiple sclerosis, microbiome in COVID. You can certainly see this moment that the microbiome is happening with, and I think this is the moment that we should be really poised to trying to be taking this to the next level and thinking about this a little bit more, but also thinking about it carefully. It's also important when you see something like this that the hype isn't outpacing the rigor of research that comes along with it. So as exciting as it is, it's also this moment where we need to be very thoughtful in how we test this so that we're not getting too ahead of ourselves here, and that when it comes to offering recommendations for our patients, we're on the level here and conducting this research with the rigor that our patients deserve, so that it's not just nutritional quackery, as I was alluding to in that paper I had shown from the 1980s. And so first, what I'd like to do is just discuss the microbiome and sort of make sure we're all on the same page here when it comes to some of the terminology that you might see in this talk and maybe in the future as well. So that's just a little cartoon here, but the microbiome is a community, not just of bacteria, but other organisms in our gut, viruses, fungi, and even parasites. There are many different microbiomes that surround us. This almost sounds like the Force in Star Wars or something like that, so bear with me here. Although there are many microbiomes that surround us, it's the gut microbiome in particular that I think will interact with the host immune system, or maybe prime it a little bit better, because our food, our intake is probably one of the #1 ways that we interact with the microbiome, the things that we put in our mouths that work our way through the GI tract. That is probably #1 in those encounters. So it makes sense that we have some of the strongest immune organs and immune lymph nodes and immune cells that surround the gut and our mouths and everything around that point. It's sort of designed, our bodies were designed that way to communicate with the rest of the world, and we have evolved immune systems to relate to that. So you can certainly imagine it's not too far off to think that the microbes that populate this environment probably influence the immune system. They do that through a few ways. Some of it is just the function that they do. They break down foods that our own genes wouldn't be able to break down, our own organs wouldn't be able to break down. So in a way, they extend the functional capacity of our own gene makeup, which is, again, kind of cool. We're more than just what our, genetic or chromosomes that were given to us by our mom and dad. And I kind of think that that's neat. We're stronger because of our microbes that are within us. They produce metabolites that can help serve as building blocks for us as we make more cells and organs. They modulate the immune system. They regulate a couple of different B cells and T cells, immune cells in our immune system. And all of this to say it's starting to be conceptualized as a new immune organ, for lack of a better word. And so one measure that comes up a lot is this question of gut microbiome diversity. What is a diverse microbiome? What does that mean? And then how can we think about this moving forward? There's a couple of different measures of gut microbiome diversity. And I think the main illustration I'd like to show here is we kind of compare them to different environments. You can imagine if your microbiome and your gut is a swamp, forgive the analogy here, a diverse swamp in the wild wouldn't be just full of alligators, but it'd be an ecosystem. It would have good fish in the water and frogs and snakes and lizards, and it might even have larger jungle creatures like, tigers and things like that. And that would be, so you can see there's all kinds of different species. They have different roles and niches in the system, and they kind of interact with the balances, and they're well represented between them. You can imagine and compare that to the swamp that maybe doesn't have fish and snakes and stuff, and maybe just has alligators. And you can imagine that's clearly less diverse. And I also point this out because as we measure this, it's not just species richness. You can imagine there's more than one different type of gator. There's alligators, there's crocodiles, there's all different subtypes of them. And you could say that if there are all those different species that were present there, they all count as individual species. But you and I know they're pretty similar to each other. And so if it's 5 different species, but they're all alligators, that's very different from 5 species where there's birds and alligators and tigers and fish and cats. So these are some of the tools that we use to measure this. We want to make sure that diverse microbiomes don't just have unique species numbers and the higher species of numbers, but that they are unique and that they're evenly represented. And there's not just token members of those species that are otherwise dominated by a certain species. So that's some of the terminology when you hear about diversity. Beta diversity just kind of directly compares between these two environments or between two microbiomes, between one patient and another patient. So there are different targets when it comes to the microbiome that we use when we're writing clinical trials for this. I will say right now, this is an area of active research, and we don't really know quite yet what the key features of the microbiome are to target. Not to mention, we don't necessarily know what the time points are that we want to target the microbiome. Does the microbiome need to be fixed forever through somebody's journey with multiple myeloma, or can we optimize it at key moments of their life? And we'll talk about this a little bit more. Is microbiome diversity what we were just talking about, the endpoint that matters? Well, I will tell you guys this, it's better characterized and it's associated with survival, which is the end-all be-all of endpoints that we think about when we're writing our clinical trials. If drug A improves overall survival compared to drug B, that is probably a future of medicine as long as it's well tolerated and safe. So microbiome diversity is better characterized and associated with a variety of oncologic diseases and settings. Sorry, bear with me, my light just went out. But hopefully you guys can still see me okay. The problem with diversity is it may not be perfectly reflective, and it might be that we can still improve cancer care by just changing a few microbes in the community, in your microbiome, without changing the diversity of it. And a diverse microbiome isn't necessarily a healthy microbiome, at least not necessarily. You can have a diverse microbiome, but if it's full of mostly pathogenic bacteria, that might not be what we're hoping for here. So is it not diversity, but is it specific populations? There are some species that have been independently associated with reduced relapses. Eubacterium, for example, was associated with reduced relapses for multiple myeloma patients that underwent a special kind of therapy called an allogeneic stem cell transplant. That's an uncommon therapy for myeloma, but still kind of points to microbiome-specific targets. Blashia species have reduced mortality from graft versus host disease in patients that undergo allotransplant. A specific bacteria called Prevotella heparoanalytica is associated in a mouse model of multiple myeloma to drive and influence myeloma progression. What about not what's present, but what they make, the metabolomics, the metabolites that are produced by the microbiome? This provides a readout. This is the function of the microbiome. This is stuff that they're making. Is it the metabolites that are made that matter more? This is One metabolite in particular has garnered a lot of attention, short-chain fatty acids, and butyrate is an example of a short-chain fatty acid. It's been associated with anti-lymphoma activity. It can help regulate and influence the immune cells that are present in our bodies. And is the endpoint what matters here, or is it the therapy that's used along the way? Is it just trying to improve diversity, or does using an endpoint like, or does using a process like a prebiotic or a probiotic, which we'll go into in more detail, matter more? And how do we reach these endpoints? Is it something like reducing antibiotic exposure as an example? And so how exactly does the gut microbiome influence oncologic disease states? I will also mention this is something that's being actively investigated, and I don't know that we have the answer yet. It might be specific for specific cancers, but one common scenario that we're using to discuss this is sort of called the common ground hypothesis. And what this shows is healthy people all have some genetic susceptibility to multiple different disorders. And that's why some people probably get cancers and why other people don't. If you've heard this before, it's probably a combination, not just of genetics, but environment as well. And so some people are more just disposed to developing these diseases. And we don't know exactly who all these people are yet, but this is an area of active research. People that have this susceptibility, maybe over time, some other hit has happened to them, a non-specific environmental trigger, maybe a chronic infection, or maybe in particular here, an unhealthy diet. This has led to a shift in their microbiome to support more dysbiotic or sort of pathogenic microbes in there. That has in turn led to an unhealthy intestinal environment that leads to intestinal inflammation, and possibly even leaky mucosa. If you've heard this term leaky gut before, it's potentially possible that this has sort of created a conduit that adds to inflammation and might contribute to disease pathogenesis. And so this combination of genetic susceptibility and then an environmental trigger leads to a polygenic disorder. So it's not one gene or one environment that can do this, but in the right set of conditions, a disease can occur. And the way that we can study this is use a genetic model of this, a mouse model here, and take that microbiome that was developed in someone that has this disease and do a fecal transplant that microbiome into that susceptible mouse model and see if we can reconstitute that disease. And if we can, that really tells you that there is a strong correlative connection here and that the microbiome matters. And so this is the common ground hypothesis that some of us try to base our scientific questions off of. And so not to get too technical here, but bear with me. There's a couple of different mechanisms by which this can happen. This cartoon at the top here shows the gut lining here. And this is the bacteria kind of in the middle of our gut where, things are in transit. And this is kind of in our bloodstream and in the skin membrane underneath the mucosa of lining our intestines and where some environmental interactions are happening. And this is where the good bacteria could be producing these short-chain fatty acids that lead to different immune activation. These immune cells then trigger and stimulate the immune subpopulations that lead to different activations that drive local immunity or even systemic immunity. These immune cells don't just stay in our gut. They hitch a ride on the bloodstream and then go and signal downstream of that. And that's why I think that this will have some power beyond just the local gut environment. This has been demonstrated, there's a couple different ways, not just activating T cells, but molecular mimicry. Sometimes the microbes that exist in our gut can resemble what a cancer cell can look like too, and that can train the immune system become more active in targeting it by mimicry and by looking like that. And so melanoma, for example, and PD-1 inhibitors, which have revolutionized the care for melanoma, have been shown different microbiome environments have been shown to differentially activate dendritic cells to lead to different outcomes when they're used to treat that. Multiple myeloma, I'd highlighted that this T cell influence that can happen. And I'll go into some more details here in a second. Butyrate and short-chain fatty acids have been related to lymphoma. And so butyrate is this short-chain fatty acid du jour here, and it does a few different things. Microbiomes, certain species in the microbiome will directly make butyrate, and butyrate can go on to influence the skin, the cell that lines the mucosa here to improve microbiota diversity and reduce inflammation. It can activate the immune cell that triggers other immune cells to improve diversity. And it can also directly stimulate T cells and change their activation. And so it's pretty active here. And if it is, you can certainly work downstream to think about influencing what makes this, which is the microbiome. And so why might diversity matter here? This is just an example of how this could matter. So this was studied using a drug that's similar to one of the drugs that we use during stem cell transplants for myeloma. CTX is cyclophosphamide, which is kind of a cousin of melphalan, which is the name of the chemotherapy that we use. And this experiment took mice that were just raised in specific pathogen-free conditions. So they had an intact microbiome. They had intact microbes in their gut. They just didn't have pathogens that were felt to be not beneficial. And they studied this in a cancer model and observed the tumor growth on that mouse cancer model when they gave just a water, when they gave just a control that didn't have chemo in it, and then when they gave that chemo. And as you can see here, when they got chemo, the tumor shrunk. When they repeated the same experiment in germ-free conditions, so this is mice that have never had a microbiome now, and don't have, they're raised in pathogen-free conditions, so they don't have a microbiome, they don't have any bacteria in their gut. The effect of cyclophosphamide, it still worked compared to not giving cyclophosphamide chemo, but you can see here the effect was attenuated. It didn't work nearly as well as if you had an intact microbiome. This suggests that the microbiome and the priming and the education that the microbiome provides to the immune system might have a role here. And when they repeated the same experiment, but then grew it in this pathogen-free condition here, but in mice that had a microbiome, but then targeted that microbiome by damaging it with antibiotics, they again lost that effect here. Again, suggesting that the microbiome might matter, and when we give antibiotics, we could attenuate and damage the microbiome, and maybe that matters in treating cancers. This is just a mouse model here, so you certainly can't take that to the bank when it comes to understanding what happens in human beings, but it certainly is worthy of more attention when you see this. And so they traced some of this to translocation events, where the gut was kind of the bacteria in the gut was translocating into immune organs that were surrounding that local gut environment and helping prime stronger immune responses than could be produced by just giving chemo alone. And so I want to pivot a little bit because I've mentioned some immune therapies when it comes to myeloma, and some of you might be familiar with what we call chimeric antigen receptor T cell therapy, or CAR T cells. But to those that haven't, this is a revolutionary immune-based therapy that takes patients' own cancer cells, collects them, and then programs these T cells, manufactures them to target a specific marker. And these are approved therapies for multiple myeloma. These are then multiplied and grown up at that company before being shipped back to the cancer center and then infused into the patient, where they go and attack CAR T cells. As they get injected into your bloodstream, these cells then move through the blood vessel, then migrate out of the blood vessel into the local tumor environment, where they activate, and they kill those cancer cells, which then has this kind of fortuitous cycle that can be created, where they release new cancer antigens that prime your immune system to target your cancer itself, and kind of in this self-fulfilling way help bring the rest of the immune system into the fight as well. And so CAR T cells have revolutionized therapy for multiple myeloma. What I'd like to serve as an example here is what I'm showing is here in 2018, one of the new kids on the block, a new standard of care that was going to improve and does improve outcomes in multiple myeloma was a drug called selinexor. And some of you may have received that or have had family members that have received this. And this kind of was the standard of care to treat advanced multiple myeloma if your cancer stopped working and responding to other therapies. So this was so good that it was published in one of our top cancer journals because of how good it worked. And this was in 2018. And as you can see here, about 50% of people it stopped working on after about 4 months or so. So, despite the hope that this was bringing, you could certainly see how it didn't work well enough for most people. And when it did work, it only bought people on average about 4 months. So just give you some context of how we were doing in 2018. Now, flash forward to 2021 when CAR T cells were approved. And there's a couple of different CAR T products, and I don't mean this is a trend that is observed for both CAR T cell products. When we gave CAR T cells here, this is an example for cilta-cel that was published in 2021. What I want to show you here first is how different that curve looked compared to the one I just showed you, where that curve was crossing 50% of people progressing and having their cancer grow. Here, it hasn't even happened yet. And then when you look at the scale, this was happening in 4 months right here with when we use selinexor. And here you can see it hasn't even crossed the midline yet. So just suggesting the power that CAR T-cell therapy has in multiple myeloma. This is just an example of extrapolating that curve that I just showed you here. And selinexor, I'm not necessarily knocking selinexor or anything like that. I think it's a drug that certainly has helped and benefited patients with myeloma, but to just show the impact and the power that CAR T-cell therapy has and why we get excited about this and why you can understand we're looking into trying to, if CAR T-cells work, how can we make them work better? I think part of that could be through the microbiome, and I'll spend some slides kind of discussing that a little bit more. But I wanted to just highlight how, we'll skip through this, sorry, for lack of a better word. I wanted to highlight this figure here. You had mentioned me talk a little bit about short-chain fatty acids. This is data here about how short-chain fatty acids can make CAR T cells work better. I just showed you how well CAR T cells work. Here, this was, again, another mouse model. They took a sort of mock here that didn't have any CAR T cells, and you can see here the tumor is growing in this mouse model. When they injected CAR T cells, you can see that this worked a little bit better than just the model here. But when they took CAR T cells, incubated them with short-chain fatty acids that are created from the gut microbiome, and then infused them, that worked even better than giving the CAR T cells themselves. This is a good example of how you can build some momentum here. If short-chain fatty acids are made from the microbiome, if the microbiome can be adjusted and influenced by things that we eat and stuff like that, and if the products of the microbiome can influence CAR T cells, it's natural to see how these things can relate and how we could use nutrition-based interventions to help guide therapies or study them moving forward. And so this is just another example of some of the immune cell populations that are driven and activated by the microbiome. These are some so-called mucosal invariated T cells. And what I want to show here is that there are microbiome populations that correspond to programming and nourishing higher amounts of these T cells, and that when you have more of these, you had a survival advantage in this figure here, which was involved patients that were undergoing stem cell transplants. And this related to a microbial metabolite, riboflavin, which is a natural vitamin and something that the microbes in our gut help create. And you had more riboflavin, you had more of these cells, and you had better outcomes. So it goes to show that there are microbiomes here that maybe produce more riboflavin in particular. And it's natural to see how we can start to think about designing clinical trials that can actually better directly address this kind of question. And so when it comes to microbiome and multiple myeloma specifically here, well, there's some research that suggests that microbiome communities drive multiple myeloma progression. This is shown in the gut here. These little microbes here are something called Prevotella heparolytica. And the presence of this microbe in a mouse model directly stimulated this T cell immune population. These T cells didn't just stay in the gut, they moved into the bone marrow, which is sort of the nexus of where multiple myeloma is grown and formed. And here, they influenced these myeloma cells and triggered them to progress and grow a little bit more than if they didn't have it. So another direct example of the ways that the microbiome can influence myeloma progression outside of the gut. Microbiome diversity, we mentioned some of these endpoints before as I was kind of defining them. If you had a higher gut microbiome diversity at the time of your stem cell transplant for multiple myeloma, you did better than if you had lower diversity, not just in length time before cancer progressed, but also in overall survival, suggesting that microbiomes might matter in the context of stem cell transplant for multiple myeloma. But what contributes to low diversity here, and how would this diversity matter at this endpoint? Well, I think some of this might have something to do with responses and might do with some of the care that happens during stem cell transplant. This is sort of some of my own research here that demonstrated the microbiome is at its worst point, its least diverse point, right at the time of stem cell engraftment, which is about 10 to 14 days after your transplant. And anyone who's been a recipient of this knows how you feel at this time. Might be experiencing a lot of diarrhea. You might be on antibiotics to treat this. You can also see that this recovers by about day 100. So if we've observed that low diversity matters, and if it's recovered by day 100, then this engraftment time point might be the one that matters most here. And so what's happening during stem cell transplant? Well, there's a lot of things that are happening, but one of the things that we observed is that if you received IV antibiotics, you generally had a lower microbiome at this point than if you didn't. Now, most people will need antibiotics. That's a supportive care treatment that has improved the lives and the safety of transplants by treating that. But it has some unfortunate side effects, and one of them could be the effect on the microbiome. So the take-home message from this isn't to not use antibiotics during stem cell transplant, but it's to kind of identify some of the factors that matter more and think about ways that we can, if we need antibiotics, and we absolutely do when we treat multiple myeloma stem cell transplants, are there things that we can do to make antibiotics safer or make our patients safer during transplant? And so antibiotic exposure post-transplant extends beyond just these microbiome effects, but we similarly observed if you were exposed to antibiotics, you had different outcomes with respect to overall survival compared to if you didn't receive antibiotics. Now, this is something that's complicated and needs a little bit more analysis to figure out, but just kind of showing at a couple of different levels from a couple of different research studies that antibiotic exposure and the microbiome may all go hand in hand in building a story about how the microbiome can be impacted and how the microbiome might impact outcomes in turn, and try and sort of create some food for thought on how we might be able to address this. And so this has also been observed not just in auto stem cell transplants for myeloma, but CAR T cell therapy as well. If you were exposed to antibiotics, you fared a little bit more poorly compared to if you didn't. And again, just highlights this role of antibiotics and how we're starting to think about this a little bit more. We observed in our own data that if you had a less diverse microbiome, of which a lot of that was driven by IV antibiotic exposure, you had maybe a not as great response compared to a complete response or very good partial response after your transplant. So maybe that response translate into those better performance outcomes, going longer, getting more bang for your buck from your transplant if you had a healthier microbiome. And so all of this to say, if we think that this is happening here, well, what kind of things can we do about it while this is ongoing? And so this kind of, again, goes back to nutrition-based interventions. So this is kind of all coming to a head here in the final minutes of this talk here, so bear with me. There's lots of ways that we can target the microbiome. There are what we call prebiotics, which is basically just a fancy term for food, the foods that nourish our microbiome. There are probiotics, which are just the felt to be key species that are healthy in our guts to begin with. Let's just put those in through yogurt or something. Antibiotics could work by reducing pathogenic species. And finally, fecal transplants, if you've heard about them, can take a healthy microbiome from somebody else and put that into somebody that maybe doesn't have a healthy microbiome at these endpoints. And these work downstream to affect the microbiome and then affect their production, potentially their immune activation, and then ultimately exerting an anti-tumor effect. And so one way that we're doing this is what I call the primal trial. And this is being conducted at the University of Nebraska here in Omaha. But basically what we're doing is taking a prebiotic and studying if it can prime the microbiome and help protect it during a stem cell transplant. I'm excited to say that we are opening a new cohort here too for patients that are undergoing CAR T cell therapies for multiple myeloma. For any of the kids or the Transformers nerds out there, forgive me, but I couldn't help but call this Optimus Primal because I called the first one primal and because we're using CAR T cells and things like that. So bear with me. But why? What prebiotic to study? Well, we chose resistant starch because it's been proven, one, it's readily available. You can just get it out of your grocery store. And so it's accessible. Two, it's been shown to improve some of those parameters, improve that butyrate production, which is that short chain fatty acid I was talking about before. And it's been studied in other people before and seemed to be safe. And so you have to start somewhere. And I felt like a prebiotic approach was the right one. It's not necessarily the only one. There's lots of different ways that we can study this. I chose prebiotics because some patients aren't quite ready to make major changes in their diet yet. So I wanted to ask the question if people could keep their same diets, but if we could just add in a nutrition supplement in the form of a prebiotic that could influence outcomes. And so we don't have data on that yet, but we've recruited to that study in auto stem cell transplants, and I'm about ready to sequence and analyze the microbiome. So stay tuned to see if we prime that microbiome differently in those that receive that resistant potato starch. I also wanted to highlight work that's supported by HealthTree and led by my colleague and Dr. Urvi Shah at Memorial Sloan Kettering. She's looking at diet-based interventions in smoldering myeloma and randomizing people to a plant-based diet or placebo, trying to see. Dr. Shah has shown preliminary data that sometimes nutrition-based therapies alone have actually taken that M spike that might be rising and bent the curve back down. So she's trying to test this in a larger setting, and I'm really supporting of her work here. I think it's asking a really meaningful question for our patients. So wrapping up here, there's a lot of different ways to target the microbiome, and some of them could be immune foods. And what I wanted to show here is a high fiber diet can be an immune food. And if people that took high fiber diets, they saw some more personalized immune responses that were changing, and that was affecting microbiome function. And if you had a high fermented food, so fermented foods like yogurt potentially, or kimchi or kombucha, they actually led to increases in microbiome diversity and actually some immune effects by reducing some inflammation. So these high fermented foods could be immune active and potentially through their effects on the microbiome. Finally, there's lots of questions that come up about prebiotic or probiotic, and I wanted to show here was some research in melanoma, so the skin cancer condition here, not myeloma, but again, some intriguing data. This was data that was studied at the MD Anderson Cancer Center, and they just took their 60 or 70 some patients, had good dietary data on them, and asked if they were being treated with immune therapy, and asked if they were taking a probiotic during that time, no or yes. As you can see here, these curves are fairly similar, but they draw attention to the fact that if you had said yes, you actually were doing a little bit worse, even if it wasn't statistically significant. So they took this a step further, and they used a fecal transplant from someone who's responding, someone with melanoma who's responding and in a complete remission from their immune therapy. And they took the gut microbiome, a fecal transplant from that patient, and put it into germ-free mice. Then they added a probiotic to that, and then injected melanoma and saw how that mouse did. This curve on the bottom here in C shows how they did. The short answer is the probiotic mice actually had their tumors grow faster than the mouse that did not receive probiotics. Again, kind of raising some awareness that probiotics aren't everything that we think they are necessarily. And if they can help, they also can potentially hurt. And until we know what the best species are, it could be that it's not a one size fits all. And we need to have some context here, not just when we're thinking about metabolic health like diabetes, but also cancer health like melanoma or multiple myeloma. This curve here shows that this probiotic approach here in D actually impaired and led to reduced diversity in the microbiome. Suggesting here that probiotics, we need to be thoughtful and careful of when we're studying them. Now, this is what happened when we used a prebiotic approach. So I mentioned that prebiotics are just things like fiber supplements or the food that nourishes our microbiome. So they took a same approach here where they looked at dietary fiber intake and looked at those that were sufficient versus insufficient, meeting the ADA sort of guideline-based recommendation for how much fiber you should take in per day. Now, the first thing I want to say is most of America falls short of this. Most people are not taking insufficient fiber to begin with. So it's actually a minority of people that are getting in enough fiber. But when they were, which is a smaller group than when they weren't, they actually looked like they fared a little bit better. And so they went a step further here and saw that the group that did the best had sufficient fiber, but no probiotic intake. And if you took sufficient fiber but had a probiotic intake, it actually looked like you did a little bit worse. Then when they looked at a mouse model, kind of something similar here, where they took a mouse model, gave immune therapy, and then randomized that mouse model receiving immune therapy and injected with melanoma tumors again, and took a look at how they did. The high fiber group did the best. Their myeloma didn't grow. Their fiber intake alone was enough to control their tumor along and paired with an anti-PD with immune therapy. And when you had low fiber, you didn't get nearly as good of a bang for your buck. And this is just showing that high fiber actually created a unique microbiome compared to the one that had low fiber, suggesting again that there's this connection potentially here of the things we take in and the microbiome. And so I want to just leave with a couple of tips to take home. These are slides that are borrowed with permission from Dr. Shah. So if you've attended one of her webinars before, this may this may be something you're familiar with, but in 2 minutes, some patients will ask, well, what can I do right now to improve my fiber? Or I would love to do that, but I can't get in enough protein. Well, let's just walk through a typical diet that could be considered here. So here's a recommended daily intake of fiber, which is 30 grams. It's a little bit higher for men, a little bit lower for women. And if we look at fiber and we look at the typical Western diet, we're not getting a whole lot of fiber with an eggs and bacon and wheat toast breakfast. If we add in a little bit of chicken and some vegetables, we get a little bit more here, but that chicken doesn't provide very much. Dinner of meat and potatoes, certainly a sort of average dinner in the state of Nebraska, doesn't give us a lot of fiber. And if we are using a little bit of a snack here, in a typical kind of not that crazy average Western diet, we've only taken in 17 grams of fiber, far below our recommended daily intake. Now, if we switch that to a high fiber diet and swap out our eggs and bacon for oatmeal and peanut butter, we've got a little bit more fiber here. If we use some lentils and brown rice instead of chicken, and can keep our broccoli going, we have more fiber here. If dinner becomes black beans and corn and an avocado, and a snack becomes almonds and a banana, well, holy crap, now we have 71 grams of fiber. And as you can see here, you don't have to be perfect. It doesn't have to be this perfect diet here. This well overcompensated for the amount of fiber we should be taking in. But little changes can get us to that daily intake. And why I show this is because if the question comes up, okay, yeah, but I also need protein during my cancer care. Well, this is the same diet. And with that Western protein here, we hit 106 grams of protein, well above the recommended intake that we need of 48 to 72 grams. And if we chose that same dang diet that had all that fiber in here, well, look at how well we did with our protein intake here. We met our protein guideline here too. So this high fiber intake, and it doesn't need to be perfect, has a way of matching, getting the best of both worlds here, matching fiber intake and also protein intake. And so some dietary tips that you can consider incorporating, well, think about more whole grains. Think about more fresh fruits and vegetables. I love this tip here, and I've even tried to challenge myself with this. I don't think the goal here is to actually hit 30, but if you start challenging yourself to hit 30 different plant foods a week, you'll start to eat a broader diet. And it can be a fun little game you can play too, because you get points. It doesn't matter how many blueberries you eat. You get a point for a blueberry. You get a point for a strawberry. And it can be a fun little game you can start playing. I want to kind of conclude here by saying nutrition changes are going to be different for different people. Some people are ready at their disease stage or where they're at. Other people might have other issues going on that make this difficult. So I want to give you all and your whoever you're supporting some grace to know that it doesn't have to be perfect. And it doesn't just have to be salads and raw vegetables. You can make little intakes here that can have bigger impacts. And you have to start slowly. Drastic changes are probably not going to be habit forming. So build something in that works for you and then build it from there. And so, to summarize here, our patients are interested in nutrition guidance. And when we don't step up, there will be a void that could be filled by opportunists and people that don't truly have our patients' best outcomes at heart. Nutrition may prevent certain cancers, as we mentioned with myeloma and fruit intake. The microbiome could be the interface between nutrition and cancer here. And there are trials that are being developed that are trying to gain and study this a little bit better. And finally, some simple advice can be done here of just some things that we can swap out or some changes that we can make at mealtime that can actually improve fiber intake. And that could be key to improving cancer outcomes, potentially through the microbiome. So with that, I will thank you all for your attention to this. And I'm happy, I know we're a little over time here, but if there is some time, I'm more than happy to answer some questions.
Excellent. Thank you so much, Dr. D'Angelo. I really appreciate that. And if you are open to questions, we did get a few from the audience.
Yeah, absolutely. I've probably got a few minutes here before I apologize, I have to run and pick up my kids, but should we start with the Q&A in the chat box?
Yeah, so if you're okay, I'll just read a couple of these to you. First one just asks, why would an oncologist after an autologous stem cell transplant say to stop taking probiotics?
So that's a good question. As you saw in the talk here, I don't mean to splash cold water on probiotics, but I think we need to be thoughtful about this. And if you've seen all these things that can affect the microbiome and all these things that can affect health, they can affect, if it can help, it can also hurt. And I think it might be worthwhile to understand specific probiotics and make sure that they actually help in the disease context of a stem cell transplant. Now, the counter argument to that could be if you received antibiotics, could probiotics help reconstitute that? And yes, there is maybe some data to help there, but sometimes that's taken from people that aren't necessarily also receiving an auto stem cell transplant. They could be getting just antibiotic therapy, for example, and don't have cancer. The final part of things is why I can't answer specifically why a oncologist would do this, but one reason that could come up to mind is a doctor that does stem cell transplants is people could be at pretty immune suppressed afterwards, and probiotics have some live therapy in there. And there's a theoretical risk, even if I think it's extremely low, and I do think it's extremely low, that that could lead to infections or lead to bacteria crossing the gut lumen and into the bloodstream. That could lead to sickness. So that could be some reasons, just as some examples here.
Okay, thank you
So,
so much. Yeah. Oh, sorry, go ahead.
oh, no. Cindy, do you want to moderate the questions or should I just read off of them?
I'll just read off them. When you do need to go, I know again we're a little bit over. Just say this will be our final, and we will call it. We appreciate all of your time, so thank you so much. So next one we had is if we don't have a diverse microbiome because of past habits, how will diet changes supply the missing species?
So I think that's a great question also. I think what they could do is allow room for growth of those missing species that aren't well represented. One analogy I just heard about the microbiome and antibiotics, for example, is this was actually in a New York Times article. They kind of described it like a wildfire hitting a forest. And so when that happens, despite the fact that everything looks charred and black and stuff like that, there could be some areas that actually were a little bit better protected than you think. And they just need room to grow. And how do they grow? Well, they need their support. They need nourishment, particularly at that setting. So it could be that they are present, but just at a low level and need their specific nutrients to nourish them and sustain them. It could be that they're present at a low level ahead of stem cell transplant and starting a prebiotic or a diet change ahead of that time could nourish them and sustain them through the course of everything they go through transplant. Antibiotics might knock them down, but maybe not extinguish them. And so give enough time and give enough prebiotic, for example, potentially that could help them encourage their growth earlier than would have happened otherwise.
I like it. I like that analogy. That is a good analogy there. Next question says, how can each of us test the diversity of our own microbiome?
That is a great question, and I certainly understand the enthusiasm that comes from that. You see this and you hear about it, and you wonder how you can test it. If you Googled online, you could probably find a company that would test your microbiome for you. I would caution that a little bit because I don't know that the state of the science. These are good research questions right now, but not knowing those independent companies and their methods, they may not provide the information that we actually need. And it takes some time to sequence all of this and get it turned around in real time. I can tell you, although I can't advocate for testing for microbiomes individually right now outside of a research setting, I can say that we are trying to develop better tools to measure microbiomes in real time. One of the things that actually can do that is checking your urine. And it's not something that you can do just yet, but microbiome research can take weeks to sequence and study and build all together to get the data that we need. And that's too slow to be a point of care tool yet, where we can take that information like a blood test that gets back in a couple of hours. One thing that we are starting to look at is the urine, because the urine can provide biomarkers of what our microbiome is producing. And so if we see those biomarkers in the urine, we can work backwards and assume that that microbiome is present. And if that's something that's key, then we can use that as saying, okay, they're doing good. Maybe they've got the microbiome that we need for our study, or they've got the microbiome they need for a more favorable outcome.
Perfect. Do you still have a few more, or do we need to call it there?
We can keep going. Yeah, a couple more minutes.
Okay, perfect. Let's say, do you see a role for oncological naturopaths in patient care?
I think this is a good example of yes. As long as I think we need to be curious in this space, and I think we need to be respectful of everybody's expertise in this situation. Naturopaths can't provide, at best, they can provide, I think, adjunctive therapies to help your doctor taking care of, your oncologist taking care of you. I think this is a target-rich area that we should be exploring a little bit more. I think if your naturopath understands, the way I do this, as an example in my clinic, is I encourage supplements if patients are taking them. If patients have a naturopath, I want to hear about some of the things that they're trying, and I will advise you if I think that it's safe or not in the context of the rest of your therapy. And if I think it is, I'll tell you, absolutely go for it. Let's figure this out together and see if we can learn something about it. If it's not, I will tell you that too. And I'll also say a disclaimer of, if I think that we don't know enough and there's a theoretical chance that it could be impacting your drugs that we're using to treat you, I'll tell you. And at the end of the day, I always tell my patients that we're a team, which means we make decisions together. And I bring my myeloma expertise, but you bring your you expertise. And if you know that you will feel better feeling like you're kind of targeting your cancer care in more than one avenue, then I'd say more power to you. And I work together on that. I'm never necessarily going to tell you no or yes. I'm just going to try to advise. And so I hope it is a more nuanced question. I think we can be thoughtful about this and stuff as long as we're respecting each other's kind of areas of expertise. And just knowing at the end of the day, I think the oncologist that's more directly kind of prescribing the drugs that have a stronger evidence base and have been more rigorously tested, trying to defer to them. But I like our oncologists trying to be more curious, for lack of a better word, and listening a little bit more, particularly when it comes to patient concerns. And that's why I think Dr. Shah has created the Nutrivention study. It's why I created the Primal trial, because I wanted to meet this sort of a hunger for more of trials that address these kinds of things. And this was one way that I felt like we could do this in a rigorous fashion.
Perfect. We've got a question about what should we eat? What would help build up that microbiome?
I think the best thing that we can do right now, I can't advocate for probiotics just yet, just as a blank recommendation for everybody because of all the data that I was showing before. I just don't think we know enough about that yet. I have yet to find adverse effects of more fiber intake. I would say it's not a one size fits all thing because you have to make it work with the rest of you and the rest of your diet. But I say eat more fresh fruits and vegetables and eat a more diverse fresh fruits and vegetables. If you're worried about getting 18 servings of spinach a day or something like that, don't. Instead, maybe the best thing for you to do is hit that 30 diverse food target that Dr. Shah had kind of mentioned in her work. Try to, and naturally, by trying to compete with yourself or maybe your partner, make it a game and see if you can get all the colors of the rainbow and hit that 30 food subtypes. You will, if you don't hit 30, you will naturally, just as a consequence of doing that, have improved your fiber intake and might have had a little bit more fun doing it. Might try some fruits in your fresh food aisle that you never would have come near before.
Perfect. I love that. Would you recommend supplements along with better food choices, or are we looking better food choices on their own?
I think, yeah, I until we know more about probiotics, I'm not. If people come into me and are taking probiotics, and especially there in some comments in the chat were describing that they're taking them for specific reasons and stuff, and so that's absolutely a reason where I'm not going to say you should stop those. It's good to advise your doctor about them and stuff like that, but I think the next evolution will be maybe that prebiotics aren't enough to move the needle. There's been some comments on the chat of, well, how does the food that we take in provide the microbe source if it's not there to begin with? And that question is well received. In addition to the other ways that I answered it, it's entirely possible that you're correct, and they don't. Now, I'll also mention that some of the microbes on the fresh foods that we eat, there are microbes on those things. So you're getting a healthy dose of microbes on the fresh foods that we eat to begin with. We're not washing those things in soap and water. But I would also say, you're right. And maybe we should come up with the key probiotic and have a food supplement that has the probiotic that we want in the system matched with the prebiotic that nourishes that probiotic. And I think that would be the next step. But it's important for us to test this rigorously before we're trotting it out for our cancer patients. And the way to do that sometimes is a step-by-step effect. And I know sometimes that's not the greatest information to hear because you want it to happen now, and I get that. But I'd say that would be the next step in the evolution here. And I'm not necessarily opposed to that approach, but I'd just like to study it a little bit more before I can just say, yes, we should be doing that.
Perfect. Thank you so much.
So
Yes, I was going
I
to say
probably
you're
need to
probably
go.
at time. Okay. Thank you so much, Dr. D'Angelo. We will go ahead and let you go and appreciate you for all those. Sorry,
Thank you all for the questions and commentary and everything. I appreciate your guys' really thoughtful questions about this and for your attention through all of this.
Perfect. Thanks again.
Thank you all. Bye-bye.
Bye-bye. All right, everyone. Thank you again for joining us today. I apologize we didn't quite get through all those questions. I will look at some other resources that we have here at HealthTree to see if we can answer some more of those questions. I also apologize. I left the raise hand option available on this webinar. The reason we usually disable it is we don't have the ability to let you ask a question verbally because of the platform that it is. The webinars don't allow us that opportunity. So I apologize for that. If you did have a question that didn't get answered, again, I'll send out some more information. And if you raise your hand and wanted to ask a question, please make sure you ask that question in the follow-up poll, and I will get those answered as well. So thank you guys so much. We will go ahead and close out today's webinar with just a little bit more information. So I'd like to take a moment to share something really exciting with all of you. One of the most powerful things, oops, I think I'm sharing the wrong screen. Let's try this again. Oh, we got it. Okay, sorry about that. One of the most powerful things about our community is how we come together to make real change happen. Right now, we have an incredible opportunity to advance blood cancer research. We're working on powering 100 research projects that will change lives. If you feel called to participate, you can contribute in two meaningful ways. First, by sharing your medical journey by joining HealthTree Cure Hub registry, because your experience matters and can help researchers understand patterns they might not see otherwise. And second, if you're able, through a donation and sharing the fundraiser with your family and friends. All donations go directly to research. In our follow-up email, you will receive the link to join HealthTree Cure Hub and to donate. Thank you all for being an integral part of our community. It is our goal to make meaningful changes in your lives through this initiative. As you leave again, please take a moment to fill out that survey. If you raise your hand and weren't able to get your question asked, make sure you add that to the survey as well. I appreciate your patience with that. Here are just a few of our upcoming events. Our Black Myeloma Health community will be celebrating caregivers by hearing from caregivers as they share their stories on Tuesday, November 21st at 2 p.m. Eastern. Tuesday, December 3rd at 1 p.m. We will have our first of many bi-monthly blood cancer financial Q&As. This is a cross-disease event, so if you have friends or families with other blood cancers, please encourage them to attend. As we've read through the comments, one of the things we've specifically seen is that our financial webinars, we request more time for questions. So when you register for this event, you'll have the opportunity to submit your questions to Diana, who will come prepared to answer those questions. If you run into any difficulties with the survey, please email me and I will pass the question along to Diana. For those who were inspired by today's presentation and want to learn more about a plant-based diet, increasing your vegetable intake, we'll be having a webinar on January 14th, all about the benefits of a whole food plant-based diet with practical tips to get started. We'll have that link to register for this event shortly. We have many more virtual and in-person events in the works that will be announced shortly. The link to sign up for these and any of the posted events will be found at the bottom of this slide and will be sent out in the follow-up email within 48 hours of the event's completion. For those who are looking forward to the webinar in January, we also would love to share one of many resources with you. Plant Powered Metro New York is offering a 21-day plant-powered jumpstart program that you can join from anywhere in the world. While the program is exciting and we encourage you to join if you're interested, there's no obligation from HealthTree to enroll. We've hosted several webinars with Plant Powered Metro New York dietitians in the past, and the dietitian from Plant Powered Metro New York will be the speaker for our webinar on January 14th. We'll send out additional information in the follow-up email. I would like again to thank our sponsors, Pfizer, Regeneron, Johnson & Johnson, Sanofi, Adaptive, and Genentech. And this was a question that kind of came up throughout the study, is getting information on your gut microbiome. So in HealthTree Cure Hub, you can participate in research studies and surveys submitted by specialists. By answering these questions, it provides researchers with invaluable information from your lived experience. We currently have a survey open, actually a study open. It's the Nutrivention 2 study. In collaboration with HealthTree, Dr. Ravi Shah is exploring the link between diet and myeloma progression. It investigates how plant-based diet and supplements can alter the gut microbiome in patients with smoldering myeloma, potentially reducing inflammation and cancer risk. The survey results will be shared with the researcher and eventually to all patients who participate. On the screen, I have the link to sign up for HealthTree Cure Hub, along with the email address of our amazing patient navigators who are available to help you create an account and sign up for the survey. I'll also include the link in our follow-up email. And lastly, I just want to thank you for all those that made it all the way to the end with me. Thank you for being a part of our multiple myeloma community. I know I met many of you and value that friendship and interaction that I get with all of you. So thank you again, and I hope to see you all again soon at another one of our events.